find a seat. We'll get started in just 2 minutes. Running on Miami time. It'll be right in a few months. I can't do it. That'd be great. All right, if you're still grabbing food, I'm going to ask you to do so quietly so we can get started. We got a jam-packed day full of events. I hope you're going to be able to join us for all of them. So welcome to our Graduate Research Day. This is one of our signature events here in the department. This is the 14th Graduate Research Day we've held. So along with the Undergrad Research Day that we have in the fall, this really showcases the excellent work that we do in the department. As Graduate Program Director, I'm particularly excited about Graduate Research Day, and it's really been amazing to see how our graduate program has grown really over the last decade. We are a very mature, research active department with research expenditures that keep growing, and that is mostly due to our graduate students. The graduate students are truly the backbone of our research enterprise here in the department. You are the ones in the lab, on the ground, doing the hard work, and we get to reap the benefits. But this is our day to recognize you, to thank you for being here, and to hear about all the awesome things you're doing. For the undergrad students, I encourage you to go to all the posters later on, go to the talks in the afternoon, learn what our grad students are doing, ask them questions, pepper them. Don't be afraid to interact and learn more about the awesome things that they're involved with. So welcome this morning. We'll be talking more throughout the day. If you haven't picked up a program, it has the full schedule in. So again, feel free to join us for any and all events that you can. So I want to start by bringing up Dr. Gadabardi, who is going to introduce our morning keynote speaker.
Morning, everyone. Thanks for being here. So today we have Dr. Jessica Pereira Houston. She is a professor at New Mexico State University. She graduated from Texas A&M in chemical engineering, so we went to school together with the same advisor, so we have a lot of stories to share, and we evolved. She moved into chemical engineering. After she graduated, she went to Los Alamos National Laboratories. That's where she picked up not just the optical side of biomedical optics, but more on the flow cytometry. And from there, she went straight into being a faculty at NMSU. And she's been there for like over 15 years right now. And since 2023, she's also the president of the International Society of Advancement in Psychometry, and she plays a key role right there. And other than that, she also has held multiple positions. In the department, she has served as an interim chair. They call it a department head at their university. Beyond that, she has gone to 2 Fulbright scholarships program for faculty. One is to Brazil, and the other one was to Japan. So she did a lot, it's like for about 6 months, if I'm not mistaken. And her work, it's being funded by National Institutes of Health, National Science Foundation. She has a very active research group. She has a lot of graduate students and postdocs right now working in her lab. And she is an advocate for diversity in STEM with the nature of research that she does. And she also has focus on research mentoring other than just being a researcher and also an administrator and different roles. So today she's going to be talking not only about her research, but also a little bit about her career path and what she has done right from the time she graduated and how she has picked up many different experiences, being also the Fulbright Scholar twice. And the most recent one was about last year.
Yeah, this last year.
So, well, welcome to Dr. Houston. Thanks for taking up your spring break here in Miami. So,
Right, yeah. A nice place to be on spring break. So there is one chair here, if anybody wants to find a seat. Again, thank you, Professor Godavarty. This is just a wonderful treat to be your keynote speaker for your Graduate Research Day. You know, I did visit FIU years back. We were trying to remember when, and I was a junior faculty then, and it was helpful to do a seminar here, and I really had appreciated it. It's 2009. Okay, yeah, appreciate that invitation. So anyway, yeah, it's just a pleasure to be here. I'm gonna, as Anu had mentioned, I'm going to talk a little bit about my research, and then I'll go into some career pathway experiences, may be helpful for you as a graduate student or undergrads that are here to learn, you know, about different routes to get to where you might want to end up with your science career. And so I'm in the state of New Mexico. I'm not sure if anybody's been in the southwest region of the U.S., but we are a border region, so we do have a lot of students that are coming from Ciudad Juarez in Mexico, so we do serve a diverse population. Las Cruces, New Mexico is just the southernmost part of the state, whereas all of New Mexico is, you know, pretty diverse in terms of geography. But I'm down here, which is New Mexico State University, whereas in the north is UNM, which is University of New Mexico. So we're the state land grant institution. It's like an engineering school. We have a lot of ag as well, influence in agriculture. So I thought I would just go through time resolved flow cytometry, which is my research area, might be of interest to some of you or in the future. Talk about some of the research we're doing, and then of course go over some of my career and background, and then just conclude. And hopefully leave time for questions. I'm happy to answer any types of questions you guys might have. So, being that this is a Coulter seminar series, I thought I'd bring up one of the quotes that is commonly found on the Beck and Coulter website, because Coulter, the development of the Coulter principle is usually known to be kind of one of the things that mostly spun or developed, was kind of the seed for flow cytometry, right? And so this is interesting when, you find this on the Beck and Coulter website, which is a huge company obviously by now that develops flow cytometry. But they say his method is one of the first, viable basis for the idea behind flow cytometry. And so now flow cytometry is just a huge industry, and I think hopefully most of you have heard of flow cytometry. I don't know if you've had. I can just do a little nutshell overview of what it is. But these all instruments here are different types of flow cytometers, right? They look, small benchtop instruments you find in your labs, clinics, and hospitals, kind of come in different shapes, colors, and sizes. But I thought it was appropriate because, we, in our industry as well, in our cytometry fields and in the field of science, we really care a lot about, the history behind it, which is the Coulter, which kind of spun from many things. So here's just the nutshell description of it. And by now, cytometers actually have become quite more complex, but this is kind of the basics of how it would work. So you have a laser source that excites samples, and the emission of light and the scattering of light can be collected by different optical detectors. The principle only works well if you have a sample that you want to interrogate and measure, such as a cell or a particle of some sort. And the measurement of a cell is possible if you can stream cells in a nicely fine single file line and separate it out. So normally the word flow comes from the fact that you were taking cells in a fluidic fashion and passing them using hydrodynamic focusing through the path of the laser beam. But when this first was invented, it was recognized right away that you can not only collect one type of light scatter from a cell as it's flowing rapidly through the path of the laser, but you can actually start collecting fluorescence. And then the next step was fluorescence of different colors, right, and different wavelengths. So any type of cytometer you might see now is considered what we call multi-parametric analysis. because for any given cell you're evaluating, if you're labeling that cell with a particular fluorophore, and you have multiple labels on that cell, you could potentially measure all of many, many different colors as long as you have the right range of spectral analysis to begin with. And so flow cytometry now, like I said, has evolved to imaging systems and so forth. But I always like to use this little video to kind of give you an idea. Cytometry is considered high throughput, meaning that cells are counted and analyzed at rates of thousands per second. So typically that's the throughput. So they're passing through a beam on the order of 1 microsecond or a few microseconds as a time scale. So it's it's a laminar flow velocity, but it's still fast enough. And in fact, this is not a really good video, in fact, because it's portraying the laser beam to be smaller than the cell, but actually the cell is smaller than the width of the laser beam typically. So so the primary reason that a person would use flow cytometry is really a clinical application. So it is a true clinical tool. So we can get true diagnostic information off of a flow cytometer. And one of the main purposes is to evaluate white blood cells, and to understand the depth of heterogeneity within a white blood cell population, to understand the levels of activated T cells and the numbers of B cells or natural killer cells and all these different cell types. So I like this image here. I find it useful to kind of just portray the diversity in our immune system. So of all these different sub-cell types, the monocytes, granulocytes, erythrocytes, eosinophils, all of these different cell types are useful in our bodies and are circulating constantly and are needed for when our bodies are trying to fight pathogens of some sort, or when our innate immune response or innate immune system is responding. There's also immune disorders that occur that changes the diversity of the white blood cells, and also cancers, for example, leukemias and lymphomas. So what cytometry has done for the field of immunology has been very, very impressive, because anytime a sample of white blood cells are analyzed on a flow cytometer, you can actually determine a multitude of heterogeneity of all of the different types of lymphocytes and other cell types. So as the state of the art today is such that you could measure 40 different colors for a label of a blood cell population using flow cytometry, and you can do it very, very precisely and it's reproducible, and it can be used for the clinical assessment. So this group with Joanne Lanigan and Maria Hymas from one of the companies, the cytometry companies, published this OMIP. It's called an optimized immunophenotyping panel, and showed that you can actually do this. So imagine labeling 40 fluorescent labels in a population. So the thing with cytometry is that because it's just like any biomedical instrumentation, it's a field that continues to grow and advance and improve. Well, in flow cytometry, because people are now trying to continue to push the limits of how many colors can be detected, it turns out many other technologies have spun out of that and to try to address that. In fact, now we have instruments called full spectral flow cytometers. So as opposed to just looking at a single wavelength with the photodetector, now we just use the whole entire spectrum. And using prisms or gratings, we put the light across an entire camera array or APD array and measure the full spectral. Now cytometers don't just collect fluorescence as a parameter, like an intensity value, but you can actually now collect an image. So just like a microscope, it's a high throughput technique that you can measure imaging, you can do imaging with. In addition to that, cytometry, it's now been about 10 years, there was a really invention of the use of mass spectrometry with cytometry. So we call this mass cytometry, so that was something that came about. Commercial companies making the fluorophores and synthesizing it. The chemists started making better and brighter fluorophores, which helped advance the field. And then there now have been a multitude of data science and AI approaches where it's needed, because now you have this high dimensional data sets that you need to deal with. And so one of the ways that we thought we would proceed with years back was to continue to try to help the field in that we want to also be able to detect all of these different colors and do this in a multi-parametric style. Is one of the ways we started doing this was to consider that you can use a time resolved parameter with flow cytometry. And to be honest, the invention of time resolved flow cytometry started even before, before my time. It started back in the 80s roughly, in flow cytometry specifically. But what essentially is the idea here is that you'd use flow cytometry, but instead of just measuring the brightness of a signal or the light scatter, instead you take the hardware and modify it so that you can measure the fluorescence lifetime. And as you know, probably most of you, maybe some of you do in fluorescence or you're in optics area, you know that the fluorescence lifetime is the average time a fluorophore spends in its excited state prior to going to its ground state. So it's a time-dependent parameter of an optical signal, and it's an important parameter because this value, this lifetime value, which typically ranges on the order of like, for organic fluorophores, 1 nanosecond-ish or half a nanosecond to maybe 20 nanoseconds, that parameter can be a very useful metric. In fact, it can be a very quantitative metric for any type of fluorescence type analysis. You can measure first or resonance energy transfer with it, frets. You can get structural conformational information about fluorophores. The lifetime is sensitive to the microenvironment within the cell, so it might be a useful measurement of that. And there's many different reasons you might want to measure fluorescence lifetime. So we thought that at the time that it would be useful to try this in flow cytometry. And the problem is it's kind of tricky to do it in flow cytometry. It's not as straightforward as in microscopy or spectroscopy or any other type of fluorimetry measurement. And the reason is because cells are in motion now. So now you're scanning samples or particles that are moving at high velocities and flow rates. And so therefore, how do you measure this photophysical trait of lifetime when it's doing that? And so one of the ways we do this is through the use of laser modulation, or we call this frequency domain analysis. And that is, in a flow cytometer, if you take a laser and you modulate it at a radio frequency, the signals that are obtained from fluorescence or light scatter become also modulated signals that you can use. And in the case of cytometry, as cells are passing one by one, if you digitize these optical signals and you measure the phase shift between a light scattered signal and a fluorescent signal, then you can relate that to the fluorescence lifetime, and this is what we call frequency domain analysis as opposed to a time domain analysis. And in our systems, what we need is high-speed digitizer to do that. We need a laser that can be digitally modulated really pretty easily, and then we need some signal processing to allow us to do this, and we do this by taking a Fourier transform of the sample. So this would be an example of a virtual oscilloscope, and this virtual oscilloscope is, let me get a little video here. It's this virtual scope here is from one of the data acquisition systems we have, and it's sampling cells at a rate, and you can kind of see the signals coming through of cells. So that was just kind of an illustration. So once we had the concept, and we thought, let's go ahead and push this technology forward in using lifetime and cytometry, we started thinking about what are the questions that are important to address. And one of the questions are, is what are the challenges with designing these instruments? If truly this is going to be useful for the field, what are the techniques that we need to be aware of? What are, how do we sensitively and reliably measure this? And then secondly, once you build the tool, right, then what are the applications that you need? Or what spaces can we influence in the flow cytometry field and the bioscience field, biomedical field, that would show that this is a useful tool? So it's a little bit like you have the hammer, where's the nail, and how do you develop this technology? So these are the two major sets of questions we've been addressing over the years we've been doing this in my lab. And I have a little bit of a list here where what are the different types of flow cytometry systems we've developed that incorporate time resolved analysis. And I don't have time to go through all of them, but I thought I'd kind of just capture a few of them. So in the whole scheme of trying to answer this first question, we've developed average lifetime technologies as a parameter. We developed instruments that try to probe multiple fluorescence lifetimes per cell. So for a given cell, can you capture multiple lifetimes within that cell? We've introduced the use of phasor analysis with cytometry data. We've done an approach where we take normal cytometry data that's non-modulated or non-frequency domain and measure lifetimes from that. We've tried different types of self-focusing methods on microfluidic platforms, so we've done acoustic focusing, and then other approaches such as time domain flow cytometry or sorting cells, cell sorting, which is another cytometry technique, and then more recently we're trying to build an optical redox type of lifetime analysis. So just kind of as an overview, this is another trace that we would measure with the data acquisition system. So the trace indicates the passage of a cell into the laser beam and out of the laser beam, and it's a Gaussian focused beam spot size, and so that's why this shape is like a Gaussian type shape. And in addition to this, what we're trying to do is digitize these entire traces, and then when you do that, you can actually do some signal processing approaches on it such that you can measure the lifetime. And in flow cytometry, it's typical that you're running samples, and the samples are collected using a histogram. And so a histogram simply allows you to count the number of cells that have a given amount of fluorescence emission or a given amount of light scatter. So when we do this in cytometry, we'll measure the histogram, and then in real time, so this is kind of a little video just showing in real time, you can actually capture the lifetime as a parameter as a histogram, developing the phase between the two signals, or even light scatter analysis like scatter. And so these are just like population. So it's a real-time analysis, I guess is the point of this, is showing that in real time you can actually extract out. And again, this is on a rate of probably a few hundred cells per second, and not as fast as a normal cytometer, but it's just slowing it down a little bit. Okay, so once we did that and we established this technique, then we asked ourselves, well, what happens if you have a single cell and it's labeled with multiple fluorophores? So if you have this cell type, for example, and let's say you have a blue fluorophore on it, or a red fluorophore and a green fluorophore on the same. So if you want to separate them out spectrally, that's one thing, but what if you want to separate out those signals based on their fluorescence lifetime? So in one integrated optical signal, can you tease out a multi-component lifetime? And that's essentially what we wanted to do, because in general, most of our histograms are just average lifetime values. So we thought one way to do this is through signal processing, because if you take a look at any type of square wave modulation approach, if you decompose a square wave by Fourier transform or Fourier harmonics, those multiple harmonics can be used as a means to exploit higher lifetime values or lower lifetime values. So for each harmonic, there's a phase and an amplitude signal that we use to measure fluorescence. So that became kind of a new approach that we took, and we showed that by combining two fluorophores within a cell that spectrally overlap, you can actually separate out those signals, and so that was kind of the cool thing we did. One of the things, I'm not sure if anybody here has heard about, it's very common in lifetime approaches or spectral analysis techniques to do something called a phasor analysis. And what a phasor is just simply a different way of graphing optical data sets and signals. And so a phasor is simply a graph that represents a vector, where the vector here, let's say you have this value here. Let's go back to my laser pointer really quickly. If you have a vector here, then the point right here represents the sample that you're measuring. The length of that vector is equivalent to the modulation of the optical signal, and the angle relative to the x-axis is representative of the fluorescence lifetime. And so, the fluorescence microscopy community does this very well, actually. The phasor analysis became a really useful tool for them to be able to separate out multiple components of lifetime per signal. And what you do in a microscope is you have a multi-pixel image, and for every pixel of that image, you can plot out the phasor graph showing what the value is of the lifetime, its modulation, as well as its phase angle. And so when we thought about doing this flow cytometry, we don't have a multi-pixel image. We just have a single optical image, bulk image from a multiplier tube or a photodetector. So every dot on this graph represents a cell. So for every cell that's being measured, you can get a population of cells that have a single component lifetime, a population of cells that have another single component lifetime, and then if you get a population in the middle of those 2 points, then that indicates a dual labeled cell. So it's a really convenient way to say, oh, I have 90% of cells with a single fluorescent label, 50% here or whatever. You can get a really nice statistical analysis of the distribution of lifetimes per cell. So this is called a phasor analysis, and we were able to demonstrate that in cytometry, which was a unique approach at the time, and also to do that as a high throughput. One of the other things that we did, again, and this is not in any chronological order, what I was just hoping to do is kind of give you an overview of different techniques we've done. But there was a point in our lab when we were starting to try to push the technology to the commercial, right? We wanted to work with companies, our cytometry companies interested in this. What are the drawbacks for implementing it, And one of the technical drawbacks was that it was a little bit cumbersome to have all of this added hardware, and you look at the pictures of the cytometers I showed at the beginning, they're these small little compact systems, right? What if you add a signal generator to it, right, to modulate a laser? What if you have to, add all these high frequency pre-amplification systems after the photodetector, which adds a lot of high frequency noise? What if you're adding all these bits and pieces to a cytometer? It makes it not as commercially viable. So at the time we thought, well, is there another way to measure fluorescence lifetime without all of this type of hardware? And the thing that we did was we said, well, in cytometry, a cell passes through a laser beam over time, right? It takes its time to go into the beam and then out. That signal is measured optically over time. So what if we actually compared the peak-to-peak delay between a cell going into a beam and out, and compared the peaks between a scattered light signal, which is nearly instantaneous, and a fluorescent signal, which takes some time owing to the fluorescence decay process. So we started on that process and tried to digitize signals with a high digitization rate, and tried to do something we call peak-to-peak, or average lifetime per cell without laser modulation. And it turns out that that was actually a very easy way to get to an average fluorescence lifetime without adding all of this frequency domain hardware or time domain hardware to any given cytometer. So I like to go around to tell cytometry companies, you can measure lifetime just with what you have there, because essentially if you're digitizing signals from a flow cytometer and you're literally measuring the peak-to-peak delay here, then you can do that. So we showed it just by doing different types of fits on the signal processing. We did like a Gaussian fit or a half area fit, and these histograms simply show that you're able to discriminate different lifetimes from the particles that we're measuring by comparing these two peak values. So one of the last approaches that I want to show you with the technology is a kind of a newer technique we're using. And this actually comes from a group that I worked with up at Los Alamos. And so what they did is they thought, well, we know that particles need to be aligned with hydrodynamic focusing, lots of fluid. What if we can get rid of all the water and fluid that is involved in flow cytometry? Instead, focus cells in single file, so carefully have cells passing through a beam without that. And one of the ways to do this is with acoustics. And so if you know a little bit about acoustics, you can use a piezoelectric transducer to send a standing acoustic field across a microfluidic channel. And when that field resonates through, there's regions, we call them nodes or antinodes, where cells will start collecting, and it's all based on the density mismatch between the cell and the surrounding media. So if you acoustically focus cells on a microfluidic platform, then it becomes a really nice way of controlling how cells passing through a laser beam. So we worked with that group, we got a hold of one of their chips and decided we're going to use this acoustic focusing approach because one, it'll give us a little bit better control for cells, because at the time we were also demonstrating that it was really hard to measure signals for reliable lifetime analysis because we were photon starved. So like if you don't have enough light, it's not as good as a reliable method to measure lifetime. So we thought, what if we can control cell movement with acoustics? It might slow down the flow a little bit, but you could do that in a little bit better fashion. And so that's what we ended up doing. And so here's a little bit of some of the hardware we have in the lab. This was during COVID, but I was with a former postdoc there. He was working on this acoustic system, and all of these are just different examples of images. I thought it'd be nicer for students to see photos rather than a lot of other stuff. So before I transition to the next part, which is the application space, I just want to give a shout out to a group I worked with in Japan, and they published this paper recently. I worked with them on it, so I consulted with them. It's not really all of my work at all, but they were very interested in developing lifetime technologies. It turns out this group is very good at imaging cytometry, and so they developed techniques where you can, at thousands of cells per second, capture images of cells. And they had this really cool idea that if you do a frequency domain approach, would you take these little beam arrays up here? So I'm going to back to the computer. If you take a dual beam array here, and for every little beam array spot, you have different frequencies that range up to high frequencies. And essentially, as a cell's moving through here on a microfluidic platform, you can get modulation frequencies up to however many beam array spots you have, and then recreate a phase versus frequency diagram to get you more lifetime analysis. Not only that, but you can capture images, so you can get the lifetime images of cells at this throughput. And it's a really cool approach, and I think that, ultimately they're trying to probably commercialize it, but I just wanted to bring that up because this is part of, a later part of the talk, which we'll talk about, working with people and collaborating, because that's really the biggest way to help you become holistic in your career. Okay, so how have we used these technologies, right? What are the biological problems we're trying to address? What are those things we're trying to solve? So this is trying to answer question number 2, right? What are the applications? And again, I won't go through every single application here, but we've done various things, such as studying protein movement with cells with cytometry. We've looked at protein conformational changes, enzyme activation within cells. We've tried to correlate the lifetime to quantum yield for near-infrared fluorescent protein development, measuring intracellular pH changes, and measuring metabolites in cells. So these are various applications that we've done. And so just as a highlight, a couple of them, again, I won't go through every single one, but I think most of us here might be familiar with FRET, which is sometimes called fluorescence resonance energy transfer or first order resonance energy transfer, when you have two fluorescent molecules that are in a close proximity and positioned the correct way, that they can engage in non-radiative energy transfer, and if you excite what we call the donor molecule, you get emission of the acceptor molecule, and you get a lifetime shift of the donor. And so we thought this is perfect for us because we measure fluorescence lifetimes, so can we do this in cytometry? So one of the things we did was we worked with somebody in Japan, again this was going back to my travels in Japan, and she had this really cool fluorescent probe where she linked a GFP molecule to an Alexa 546 fluorophore using a peptide sequence. And when you introduce that into the cell, and the cell then is initiated, apoptosis is initiated, enzymes occur during that process, and one of the enzymes that becomes really active is caspase. A number of different caspases, but in this case we were looking at caspase 3, and it cleaved that peptide, releasing the two fluorophores from each other. So you either have FRET, which is energy transfer occurring, or a loss of FRET, or energy transfer. So we were able to show this in the development of apoptosis through the caspase activation over time with cytometry. So what's the benefit there? I think the benefit was that we showed that you could do this at a high throughput with hundreds of thousands of cells, as opposed to just in a microscope where you're looking at 10 cells or 100 cells. We also did a FRET study with another group at University of New Mexico. They had this really cool FRET approach where they were looking at integrins. So integrins are expressed on the surface of cells, very important for motility, lots of other things. And so when an integrin is activated on the surface of a cell, its conformation changes to a standing. This is in particular the alpha-4 beta-1 integrin. So when that integrin's activated, they wanted to know, well, how much of it is being activated? Is it 100% across the surface of the cell? And so they designed a FRET probe where the donor was on the surface of the integrin, and the receptor molecule was in the membrane of the cell. So if the integrin was non-activated, there would be FRET occurring because those two fluorophores are closer in proximity, when there was a loss of FRET and integrins were activated. So we showed this, and this little graph here shows an illustration of how we evaluate this type of data sets. And you remember the phasor plots I showed a minute ago? This is a phasor graph, and you can actually get a FRET trajectory graph where you show the trajectory of what the donor emission lifetime is all the way to when the donor is engaged in energy transfer. And you can get FRET efficiency as well, and it's kind of like you can do a regression to evaluate that. Okay, so I think this might be the last application I'll show before I kind of get to some career stuff. And this is more recent. So this is something more recently we've been focused on. There's a long history of studying metabolism in cells by the use of autofluorescent species inside of a cell. And maybe if you're in an optics field, you really know that. Dates back to Britton Chance, who first identified that there are these fluorescent molecules, and some of them are metabolites, and that they actually are good metrics to understand when a cell is engaged in a different specific metabolic profile. And so these metabolites are known as NADH and NFAD, or NADPH and FAD. And in the field of microscopy and also imaging, it's very common to know that you can make an observation of a cell and just measure the autofluorescence from that cell, and then try to quantify what kind of stage of autofluorescence or what stage of the metabolic profile the cell is in. And so in cytometry, it's a little less common because we think of autofluorescence as a hindrance in cytometry. We want to get rid of autofluorescence. It seems it's usually a background that's kind of non-important to us. But when we started thinking about this, we thought, well, if you were actually measuring autofluorescence for a purpose, then it would be useful. And so because an autofluorescence has a specific lifetime shift, then it even became even more interesting to us because we have a cytometer that can measure lifetimes. So if you're not aware, NADH goes through a process in which it's, either during a glycolytic cycle or in the TCA cycle or in the mitochondrial respiration, it'll go through as an electron donor to either be bound to specific enzymes or unbound. And when there are complexes that bind to NADH, like lactate dehydrogenase, then that occurs a lifetime shift of that molecule specifically. So because that's the case, if you measure the fluorescence lifetime of NADH and inside the cell, of the overall fluorescence lifetime, you could make a prediction that whether or not that basal energy metabolism of the cell is going more toward glycolysis or more towards oxidative phosphorylation. And so then that became a useful thing for cancer, right? Because we know that cancer cells are, utilize a lot of energy, even dating back to the Warburg effect and things like that. So cytometry field started kind of picking up on this as a metabolism thing, and then we started looking into it because we have a tool that can measure fluorescence lifetimes. We thought that was really cool. So we started working with Kevin Houston. He's my husband. He's in the biochemistry department at NMSU, and he studies breast cancer, and he studies breast cancers that are tamoxifen resistant. Tamoxifen is adjuvant therapy for women undergoing treatment for estrogen receptor positive breast cancers, and it turns out that there are a lot of patients that become resistant to that over time. Why is that? That's a kind of a question he's starting to answer. So we started taking his tamoxifen resistant breast cancer cell lines and just scanning them for fluorescence lifetime changes to try to understand, is there a metabolic situation occurring here within these resistant populations? And we did find that there's differences between the resistant populations and the non-resistant populations, whereas the resistant populations were more likely undergoing a glycolytic metabolic state as opposed to the non-resistant. So it's a developing story, but we have some published work where we compared this to different types of metabolic analyses, like a seahorse assay. If anybody's heard that, I can answer questions later. And then in the literature, like Melissa Scales in Wisconsin, her group, and Michelle Digman, different people in this field in microscopy have shown similar trends for different types of breast cancer cell types, either triple negatives or estrogen receptor positive breast cancers. And this is just a data set from one of their, I think this is Dickman's group, where she treated with cisplatin and showed that resistance to chemotherapy is also showing a glycolytic trait. Now, I would not call tamoxifen a chemotherapeutic. It's more of a hormonal treatment for breast cancer, but there's similarities, so that's the idea here. Okay, so the summary, the takeaway is, hopefully I showed you a lot of different, potentially cool ways to do lifetime and cytometry, or ideas maybe spark your interest, and also different applications. I do have a couple minutes, and I want to talk about my career path, because I think this is maybe interesting to some of the graduate students. So I'm at New Mexico State University. That's where I actually ended up starting. So I'm from New Mexico. I grew up in Santa Fe, which is the northern part of the state. I was a student in the early 90s, thinking about college, and my family couldn't afford me to send me anywhere, right? I had to look at where was local university that I could go to, and I wanted to be far away from home as possible. So I moved 4 hours south to New Mexico State. So I started there. I didn't really know I wanted to do chemical engineering, to be honest. I grew up with an engineering father and a teacher. My mom was a teacher. And I always liked biology, and I was thinking bio, I know there's something called bioengineering, that's kind of cool. But New Mexico State did not offer bioengineering. In fact, to this day, we still don't have a biomedical engineering department, it's chemical engineering. My dad told me, well, why don't you try chemical engineering? It could be interesting. So I started there, tried chemical engineering, and ended up liking it enough to stick with it. But I got involved in research as an undergrad. So probably a lot of you did undergraduate research. Some of the undergrads that are here are doing research. And at the time, I thought, I definitely do not want to work in the process industry. I don't want to get a job at ExxonMobil or Intel or Dow or DuPont, wherever is hiring chemical engineers. That's just not for me. Archer Daniels Midland, I did an intern there. It's not that very exciting to me. So I decided to go to graduate school. And interestingly, I picked A&M not because I was thinking that was the best school for me. I picked it because my husband at the time, we had just gotten married. I was graduating from undergrad. He was going to UT. So he was going to UT Austin for graduate school. And so I thought, well, I'll apply to UT and maybe we can both be in Austin, actually. So it turns out I did not get into UT as a graduate student. My application was rejected. And so, I wasn't very strong. I didn't really necessarily have the strongest GRE scores. I was, kind of maybe a 3.7 student, but I applied to A&M and I told Kevin, maybe we can live somewhere in the middle of Texas, and I go one way and you go the other if I get into A&M. And so I did, and I applied and I got into Texas A&M, and it turned out to be the best, the best opportunity for me. Sometimes you never know where you're going to land, and it was great. So I went to A&M for graduate school, and then I'm going to tell a little bit of a story of kind of coming full circle. So, but I thought this would be a fun picture. Do you recognize anybody here? I don't know if you can find me, but I see somebody I know right here. Yeah, okay, that's me. When I went to Texas A&M, at that time, A&M was not as huge as it is now. This is the university's grown immensely. The chemical engineering program now has its own big building. At the time, we were part of a bigger institute. But when I went to A&M, it was very eye-opening. And the reason why is because I was used to my land-grant state university. A lot of people looked like me. I was, among a lot of students that I was from the state of New Mexico. Go to A&M, and it's totally international. And you guys are international. You're Florida international, but you're a diverse, group here, and you're graduate students as well. But when I went there, I thought wow, this is, do I fit in? You get the imposter syndrome a little bit. Do I even fit in? Am I supposed to be here? Because I see all these people, and they're so smart, and they, you learn a lot from them. But it was really eye-opening. And I think the challenges there is I was I felt like I wasn't quite prepared like my undergrad. I did chemical engineering, but I would go to these graduate classes and sometimes I would sit there like, I don't really know what's going on, and it was a bit of a challenge. So I have to admit it wasn't easy, but I made it through. Well, at the time, so this is our advisor, her name's Eva Sevick, and she was kind of moving around a bit, and so right before I graduated, I think Professor Godevarte graduated already, she transitioned to Houston, so the city of Houston, and the Baylor College of Medicine there. And she had a new position there, starting up a diagnostic imaging and a radiology department at Baylor. And so she brought most of the team there, and I was still there as a graduate student, almost to graduate at the time. So I ended up finishing my degree at A&M, but then living in Houston with her and the rest of her team that she had hired. So there's me with some of the other team members at the time, and I just had a great time in Houston. I mean, we lived in the Texas Medical Center, it was great. And when I finished working with Eva, she kind of kept me on for a little bit. I thought, look, I don't know what to do yet. At the time, my husband, Kevin, had gotten a postdoc at Rice University, which is also kind of in the Texas Medical Center area. So it was working out for us. We were like, okay, we have 2 career pathways here going. Kevin's in biochemistry, I'm in engineering, and we have this Houston situation occurring, and it's fun, and we started becoming Astros fans, and to this day we still take our kids back to Houston and go to Astros games. Well, at some point in your life, if you have a significant other, you start thinking about family, potentially. Not all of us, but some of us. So we started thinking about family, like we want to have kids, we want to start a family. What would make most sense for us if we wanted to start a family? Well, maybe it makes sense to go back to New Mexico because that's where we have a lot of support, right? And it doesn't work for everybody, right? Because you may have family that are in different countries or different places, but for us, we thought going back to New Mexico would be interesting or useful. So that's when we started looking at places in the state of New Mexico for 2 PhD people in science to get jobs. And the state of New Mexico is a rural state. It's very small in terms of population. There's not a lot of industry. And the major industries are the university systems and the national labs. And so we thought, why don't we try to find a job at one of the national laboratories? And I'm sure a lot of you have seen Oppenheimer, the movie, right? You know about Los Alamos National Lab. It's where the place where the nuclear weapons were developed. And it's still to this day, a lot of their mission is in weapons and the stockpile stewardship program of the nuclear stockpile. And so of course, that was a Department of Energy lab, or it's actually run by NNSA, which is the National Nuclear Security Agency now. So, but interestingly, back in the 1960s, Los Alamos was a place that was studying radiation biology. So they were looking at radiation effects in individuals. And at Los Alamos, they also had access to a lot of high-power lasers. So it turns out a lot of people were starting to use lasers for, interrogating different things, maybe in biology. And one of the things they started doing was using lasers with cells, okay? And this evolved over time. So this is my group in Los Alamos, and this was a group that was known as the Flow Cytometry Research Resource, and that was me as a postdoc, and I don't know if you can recognize any of this equipment, but this is actually a high-power laser. It's one of those lasers that's pretty as long as like this table, and it has to be water-cooled. So I started getting a lot more experience with cytometry types of techniques in Los Alamos, and started working around a lot of good people that were developing. And so a fun fact here is that Los Alamos is one of the places where cytometry was invented. In part, it took place during those years. It took place in different places in the country, but one guy in particular, his name was Mac Fulwiler, developed what we call the cell sorter. And Mac Fulwiler publish this in Science in 1965. So that's 60 years ago now, right? So that invention, the cell sorter, spun into many different technologies at the Los Alamos National Lab, a place known for nuclear weapons. And interestingly, after cell sorting was invented, the Genome Project occurred, in the 80s and 90s, and cytometers were used to sort chromosomes to do sequencing, so to do the Human Genome Project. But that's kind of an interesting history of Los Alamos. And when I started learning about cytometry there, you learn about this rich history of work. And I ended up loving it enough to take it back to NMSU. And so that's what I did, is I came back to NMSU. Kevin and I joked that we would never go back to NMSU because we met there. And when we decided at Los Alamos, we thought we could stay as staff scientists there, or maybe get a job somewhere, but where would we? Maybe we could do a faculty position. And I thought, no, I would never teach chemical engineering classes in my life. And we saw that there were faculty position openings, and we applied for them and realized, hey, this is a flexible job. This is actually a really cool job. We could create our own hours. I mean, of course, we want to be there to teach, but we can have, tailor the research we want to do to what we want to do. And, it's not so, it's kind of sounds interesting. So I started teaching and figuring out how to teach. So there's a lot of pedagogy behind teaching, and like active learning and team-based learning, and started learning about that. I started building a lab, graduating my first PhD student, started working with Kevin. Now Kevin and I consider our groups one big group, actually. We just work together all the time, which is really nice. And since then, over the years, like Anu said, I've been there 15 years now. I've just tried to develop an academic career that's kind of important for my family and for the goals that we want. And so I've done a lot of traveling with my kids over the years. I've exploited the Fulbright program, and that's definitely a program to look into. And just done a lot of different, service to science and all of that. I do have to shout out to Fulbright because I'm a Fulbright liaison now, and if anybody's interested, I can talk to you about it. But we lived for 6 months in just north of Tokyo, and then I went to Brazil to work with this guy Henning in his biochemistry lab just last year for 4 months. And so I think the Fulbright is just a great way for you as a faculty to leave, take your family or yourself to a different country. Students can also apply. There's Fulbright programs for students as well. And then lastly, it's kind of, as scientists, we sure, a lot of the things we do, I think, is a selfish thing. We want this for our research, but we also work in the context of others, so we're not silos. And so what we do to the world of science is important, right? And now think about all the stuff happening now politically, right? And it's, you wonder, what's going to happen with research funding, but what we do outside of the doors that we work in is really important. And so I've kept working with this one particular society over the years, and that's ISAC, International Society for Advancement of Cytometry. And I've kind of worked my way up through it as a volunteer, member of the society, volunteered on committees, became committee chairs, ran for council, ran for treasurer, and now I'm serving as the president of the society. And it's not a huge society, it's like 2,000 members or so. We run an annual meeting. But I think over time, it's slowly shifted the makeup of the diversity in the science, right? So before that, we wrote a paper, myself and a few other editors in our journals, our flagship journals called Cytometry Part A. At the time I was on the editorial board for that journal, there were 4 women and 23 men on that journal. We wrote an article to the editor, and since then it's completely changed, right? Now we have, now it's basically a diverse group of not only people from different parts of the world, genders, but also, expertise. And so it was really something that was important. Now there's our president-elect. This is our past president, Johnny. Sorry, she's cut off. Virginia's our president-elect, and me. Before us, it was a long history of men as presidents in leadership positions, which, kind of is, natural science things are changing, but we have a now executive committee. Our first executive committee that was all women was last year when we had a secretary, treasurer, past president, and president-elect. So I think it's important to continue to contribute, and I do, value, the diversity in science. Okay, so some of the people listed here, there's work that I didn't mention or didn't show, but I do want to try to be as comprehensive as I can. So a lot of people I've worked with over the years at UNM, at Venue Bio, the company that does acoustics, at Touch, which is people that I worked with doing like the phaser stuff. Miltony is a data acquisition company. Formerly Dark Connects, Miltony is a cytometry company. At Saitama and at University of Sao Paulo, other places, Albert Einstein. But here's my group, and again, this list is not comprehensive, which I should update, but these are a lot of the former graduate students and postdocs in the group, and this is kind of the current makeup of some of the students in the group right now. But I do want to mention that if you're looking for postdocs, I'm hiring, and also if you are a undergrad and you're interested and the research sounds interesting, come talk to me, because I'm definitely always looking for good graduate students. And then the last slide is just kind of to promo some of the things that I've been doing. One is we have a conference coming up in Denver. I know most of you are at cytometry, I think, and I've heard that maybe there's not so much in the department here that's doing, single cell analysis or microfluidics, but we are looking for, I'm also editing this special issue in biophotonics discovery. So if you're interested, you think you have science or work to submit there, let me know. And our journal is always looking for good papers. And again, that's all I have, so thank you.
Great talk.
Yeah.
Questions? Any questions? I had a question. I like the idea when you said instead of going and doing the entire frequency domain, you try to do why can't I just do by digitizing it?
Yeah.
Particularly when you talk of fluorescence, the lifetimes or a nanosecond or picoseconds.
Yeah.
Which means your ability to digitize your camera should be that fast.
Yes. Yeah.
And I don't know if the typical flow cytometry cameras that they have commercially, are they that fast in digitizing, or is it something that you post-process to figure that out?
It's a post-processing, but instead of like a camera, we're using just a photodetector, so like an APT or TPT. So at the rate at which photons are coming in, as long as that digitization rate of the DAC is high enough, then you would capture them. But there's some limitations, but
Because I know you mentioned you don't have to modify your current instrument.
Yeah.
So how does that work there?
yeah. So in that case, if you had a standard cytometer and you were able to, so normally you get only the peak of that value, but if you had the entire curve, if you had the digitized value, then you could take that and then maybe do a fitting of that, find what the peak is at that time, and then compare it. So it's
It's more than analysis.
it's more than it's more of the analysis.
Really cool talk, both sides really enjoyed hearing about the journey as well. For the imaging modalities, you talked about kind of the end with the photogrammetry. What's the spatial resolution there? Is there any possibility you could ever get to subcellular distributions or anything like that?
The spatial resolution's not there, not as much as at all as you get with like the confocal or so I don't know exactly, because it would vary based on the instrument, but what they say and what I've seen in images is that it's enough to kind of get an idea of existence of a fluorophore here, nucleus versus cytoplasm or membrane. So you can kind of, and you can get morphology, so you'll get some shape out of that. So it still suffers, and I think that's one of the reasons maybe in imaging cytometry people are trying to like figure out ways to improve it, or also make a case that, well, maybe you don't need the resolution as much as you need the throughput, but you can gate a cell sample based on like just this shape of the nucleus or things like that. But the exact resolution I couldn't say off the top of my head.
But it's still in microns.
Yeah, it's microns. Yeah.
This is a very naive question, and I probably don't know if this makes sense, but has someone tried to combine this kind of fluorescent and lifetime with polarization? Because the cell, since there's a linear band of linear polarization depends on the cell type, that someone tried to combine.
I can't think of anybody in particular that has, or that I've seen a cytometer that uses that, but I've talked to many people about that idea, and you're right, because polarization of the sample can give you maybe more rotational effects or things that are occurring. So I
Diameter of the cell,
or
thickness
right.
of the membrane.
I mean, by by nature the light's polarized, you could maybe, Los Alamos did something similar to that in the past, I remember. But it didn't take off very well, but I know it's possible, right? You think that maybe it's an approach that's out there. But I can't think of a particular, well, not commercially, but people, maybe in somebody's lab, they've done it or just demonstrated that.
One more question. You said you use a lot of high power lasers.
Yeah,
Is it because,
not so much
so
anymore.
can you just use like the simple laser diodes?
Oh, yeah. Like these days, we just use the small.
And those small, whatever frequency synthesizers, and you should still get the.
A laser pointer like this would
Should
work.
do the job.
Yeah, and as long as you can modulate it digitally or make it easy. In Los Alamos days, we did because we had to use an electro-optic modulator and external modulator because that was the only way to do it. But now the technology's
And the entire technology is invisible.
and visible. Yeah, it's visible. Or we try and we use UV for the metabolic stuff, but then we use most of the visible range for other stuff. Not so much near infrared, but
Yeah, you don't need it.
yeah. Guys?
Okay, if there's no other question, let's thank the speakers.
Quick announcements. Thank you again. So poster session starts at 10:30, and I want to also make sure that we'll do this several times throughout the day to acknowledge our amazing staff as you go through the day. Like we have by far the best staff in the university here in the department. So especially Ingrid for all the promotional work, Vanessa and Claudia for all the logistics. Absolutely fantastic. We have a fantastic day. Please join us for the poster session, 10:30 in the Panther Pit. If you are judging, if you know that you're judging, come see me. If you're interested in judging, we also could use more judges. Undergrad students, postdocs, faculty, anyone who wants to judge, come see me and I can provide instructions. All right, see you soon.